Hello, everyone, and thank you for joining us this evening for the third week of the Rugby Research and Injury Prevention Group 2026 Journal Club. We'll begin our first Journal Club presentation, and our first intern is Julia Viviani. Julia will be presenting on chronic traumatic encephalopathy in athletes involved in high-impact sports. Julia, you can take it away.
Hey everyone, let me just share my screen really quick. Let me see if it. My name is Julia Salim Viviani. I am a rising senior at Drew University, and today I am going to be presenting on chronic traumatic encephalopathy in athletes involved in high-impact sports by Cyrus Safina and colleagues, published in the Journal of Vascular and Interventional Neurology in 2016. So before I start talking about my paper, I would like to refresh our memory in what is CTE, since it has been a couple of weeks since Elon did a great presentation in CTE and she already defined the disease for us. So CTE can be defined as a progressive neurodegenerative disease caused by repetitive mild traumatic brain injury, or MTBI. Its main characteristic is the abnormal buildup of hyperphosphorylated tau, or p-tau, in specific regions, and athletes in high-impact sports such as American football, boxing, hockey, and rugby are significantly affected by this disease due to their high-impact exposure. So when I'm talking about today's paper, which is a review article, I'll be talking about their research in important CTE aspects, such as epidemiology of CTE, clinical presentation, clinical and imaging biomarkers, impact of high-impact sports in CTE development and its differences, and a couple of study limitations and the conclusion of and future research in this article. So CTE, as I said before, is associated with repetitive MTBIs in high-impact sports. The paper reports that between 1.6 and 3.8 million sports-related MTBIs occur annually in the United States, and actually, sports-related injuries account for approximately 20% of all traumatic brain injuries. The highest risk sports include, as I said before, football, boxing, ice hockey, and rugby. But most importantly, they're talking about the sports in all levels, from high school to collegiate to professional. That is important because high school football players actually sustain an average of 24 head impacts per game that exceeds 14 G-force, which is very strong. That is why recent research has been concentrating on the development of protective equipment that acts to mitigate the effect of high impact forces. So although concern about CTE has increased significantly during the years, unfortunately, the official diagnosis of this disease can only occur and be confirmed through post-mortem brain examination. In the paper, actually, the authors emphasize the necessity of reliable in vivo diagnosis criteria and how the lack of it limits epidemiological studies. So when talking about the criteria to describe CTE and how they appear in patients, the paper describes four major symptoms categories. So cognitive symptoms that consist in memory impairment, executive dysfunction, and reduced attention. Mood symptoms that consist in depression and the feeling of hopelessness. Behavioral symptoms that consist in physical and or verbal aggression, impulsivity, and mercurial behavior. And motor symptoms that are seen in more severe cases that are ataxia, dysarthria, Parkinsonism, tremor, and abnormal gait. And actually, the average of the symptoms onset among confirmed cases was approximately 43 years old. And also, the symptoms develop gradually, and most of them don't appear actually after years has gone by and after their repetitive head trauma. So to reinforce this criteria, the paper used the McKee 2013 study that proposed 4 stages of CTE progression. So stage 1 is characterized by headaches, attention deficits, and poor concentration. Then stage 2 is depression, short-term memory loss, and impulsivity. Stage 3 is progressive cognitive impairment, and stage 4 is language dysfunction and aggression. So, although many symptoms can be described as characteristic of CTE and its progression, it's important to understand and remember that diagnosing CTE is particularly challenging because many of its symptoms overlap with those of other neurological disorders. So patients may present with memory problems, mood disturbances, behavioral changes, or motor symptoms that are also seen in other conditions and diseases such as Alzheimer's disease, frontotemporal dementia, Parkinson's disease, and post-concussive syndrome. This table that is right here in this slide actually highlights those similarities, and it helps explain why a definitive diagnosis of CTE currently remains difficult during life. As shown in table 1, which is this table right here, several neurological disorders share clinical features with CTE, like on the signs and symptoms column where the headaches and depression behavior are both associated with CTE and PCS. This then makes differential diagnosis an important part of a patient's evaluation. So the paper also identifies Alzheimer's disease, frontotemporal dementia, post-concussive syndrome, Parkinson's disease, vascular dementia, and psychiatric disorders, as you can see in the table, as important mimics of CTE. However, there's a critical differentiation factor between CTE and these mimics, which is the history of repetitive MTBIs. For example, post-concussive syndrome, or PCS, usually follows a single traumatic event, and symptoms appear relatively quickly. In contrast, CTE symptoms typically start to show between 8 to 10 years after repeated head trauma. And similarly, patients with Alzheimer's disease or frontotemporal dementia usually lack a history of repetitive head injury. So, with the intention of trying to find a way to diagnose CTE in vivo, these authors discuss ongoing research to identify biomarkers. The most promising are the CSF biomarkers that include p-tau and neurofilament light biomarkers. Actually, a study on Olympic boxers shows that p-tau increased shortly after boxing matches, but returned to baseline after recovery. And on the other hand, neurofilament light polypeptide remained elevated longer, suggesting that it actually may be a more useful marker when it comes to CSF. But to get those CSF biomarkers, the process can be a bit invasive. So an idea of biomarkers are a blood-based biomarker. However, the development of those biomarkers are very challenging because proteins from the brain are present in very low concentrations in the blood. So, unfortunately, at the present, there is no current biomarker that has been validated for a clinical diagnosis. So to try to find other ways to diagnose CTE in vivo, the paper looked at imaging. In the case of CT scans and MRIs, the routine CT or scan or MRI would not show any abnormalities, and that only changed when the disease was in a very advanced stage. So to prove this idea of CT scans and findings with MRIs, the paper used these two figures that you can see right now separated in panel A and panel B in this slide. So panel A, we have a CT scan of a retired football player that exhibit substantial cognitive and behavior symptoms that correlated to CTE, yet their CT imaging showed very limited abnormalities. And in panel B, you can see that this is a CT scan of a postmortem of a professional boxer, and it just shows what would regularly be seen in a person that went through repeated head trauma. So basically, these images illustrate why conventional CT and MRI alone are generally insufficient when it comes to diagnosing CTE. However, PET scans have actually been showing a very nice potential in imaging of CTE in vivo. That is because the PET tracer 18F FDDNP binds to tau-containing structures and allows the visualization of abnormal protein accumulation. So figure 2 here in the paper, that it's right here in the slide, shows a study where patients with suspected CTE have actually a different distribution of PET signal compared with patients with Alzheimer's disease. So the CTE one is the one in the middle, and the Alzheimer's disease one is the one to your right. So you can see that because specifically there's a stronger uptake because there's like a stronger uptake observed here in the mesocephalon, amygdala, and subcortical regions. In the CTE area, you can see it in the bright orange. And these findings also correspond to known pathological patterns seen in autopsies. So after analyzing the efforts to find an in vivo diagnosis for CTE, the authors proposed that actually different sports cause a different biomechanics impact, and that can actually explain the differences in CTEs in different people. So boxers, as you can see on the left, they experience more of a rotational acceleration, specifically from hook punches. And these rotational forces create a shearing-like injury that damage axons and deep brain structures. While football players, on the other hand, experience a larger linear acceleration, often from helmet-to-helmet collisions. This study actually reports that 83 professional boxers showed debilitating motor impairments compared to only 19% of football players. And therefore, this finding suggests that the type of force experienced by athletes may influence both pathologically and clinical presentation of CTE. So during the writing of this review article, the authors ended up coming with various limitations. First, CTE can only be definitely diagnosed after death, making it difficult to determine its true prevalence in athletes. Also, many studies rely on retrospective analysis of donated brains, which induces referral bias because individuals who donate their brains may not represent the general athletic population. Another limitation is that I actually said before multiple times during this presentation is the absence of validated clinical diagnosis criteria, which complicates epidemiological studies and clinical pathological comparisons. And finally, CTE shares many symptoms with conditions such as Alzheimer's disease, frontotemporal dementia, and post-concussive syndromes, which make diagnosis during life particularly challenging. So this study concludes that CTE is a complex neurodegenerative disease associated with repetitive MTBIs. And although important progress has been made in defining the neuropathology of CTE, the true incidence and prevalence remain unknown because diagnosis currently requires post-mortem examination. Also, the authors identified that the development of reliable in vivo diagnosis methods as the most important future research priority, and PET imaging and biomarker studies are particularly promising. They also recommend for future research longitudinal studies linking head impacts to disease progression, clinical pathological correlation studies, and prevention strategies and improvement protective equipment and protective equipment. So these are my reference for today, and thank you very much. Any questions?
Thank you for your presentation, Julia. I think it's really important to understand CTE, especially because you hear it so often mentioned in conversations about football, contact sports, and I guess like fighting sports like mixed martial arts and boxing. And I think it's important to understand how complex it is and how hard it is to diagnose and treat this kind of a disease. So, Dr. Lopez, do you have any comments? Does anyone else have any questions for Julia? Jack, go ahead.
I don't have a question, but I just think it's like I just want to comment that I like commend you because I think it's a really important topic. And like I was curious when you said that the in contact sports, like the average G force experienced by the players is 14 grams. And I looked it up, and astronauts shooting up in a rocket ship generally only experience like 5 to 6 grams. So in that short amount of time to experience like almost double the amount of G force can have like tremendous effects on the body. So I think it's a really important.
Yes, definitely. Actually, just a
So
comment on Jack saying what Jack just said. To think about that you're talking that amount of G-force is in high school players is actually really, really impressive.
yeah, are there any more questions? Can we move out of the presenter slide here? So I guess what I wanted to ask Julia, the amount of um people that they did the MRIs on or the CAT scans and the main studies on, how many did they incorporate or how many did they use? Did they discuss that?
They didn't discuss amounts since it's a review article. They used based on other articles, so they just basically told like the conclusions. I can look at the articles they use because they obviously
That's
tag.
fine. That's fine. No, I was just trying to figure out how many. Obviously, my question is, how did they choose that population? Did
For
they screen them? Obviously, and just relax. We're just talking as a generalized group.
of course.
You'd have to try to figure out who's preemptively presenting, or you'd have to go with a history. So I'm
Yes.
just wondering, what did they lean on most to try to figure
I
out
think
which?
what they leaned on, I think from what I read during the paper, what they leaned on most, I think it was athletes that do high impact sports more than necessarily if they're presenting like signals or like any symptoms of the disease
Symptoms,
itself.
preemptive symptoms, pre.
Yeah. I think they were looking more at athletes itself that of sports that are known to later develop CTE more than symptoms.
Mm, interesting. Okay,
Thank you,
good
Julia,
presentation.
thank you, Julia, for that wonderful presentation on CTE. Our next intern that will be presenting is Erin Zeng, and she will be presenting on drug testing and the risk of unintentional anti-doping rule violations when consuming hemp products. Erin, you have the floor.
Thank you so much. Um, I'm going to share my screen. Hi, so today I'm going to be presenting this journal article, which Hunter already introduced. It's the risk of unintentional anti-doping rule violations by consumption of hemp products, and it was written by Ut Marek and his colleagues. It was published in 2023, and it's in the journal called Drug Testing and Analysis. So first, in the introduction of this journal article, it talks about the hemp plant, and it discussed how in 1996, a prohibition on cultivation of low THC cannabis plants was lifted, and that caused hemp products like hemp oil or seeds to become popular, particularly for their health promoting effects, because hemp products can, for example, support cardiovascular and digestive health. And hemp contains over 150 cannabinoids, including um, less than 0.3% concentration of THC. And if there was any more THC in the plant, it would be classified as marijuana. And the two are actually the same plant, but because of this lift in prohibition, it caused them to be legally classified as two different things. And cannabinoids are just chemical compounds that can act on the endocannabinoid system, and that regulates functions like mood, sleep, appetite, and pain. And that can be found naturally in the body or in plants like the cannabis plant. And an important thing to note is that even though hemp has a capped concentration of THC, it can have other cannabinoids at varying concentrations. And so the in the introduction, it also talked about how the World Anti-Doping Agency prohibited all natural and synthetic cannabinoids in competition, except cannabidiol, which is CBD. And this could be concerning for athletes who consume hemp products with these cannabinoids. And so the goal of this research article was to determine the likelihood of athletes testing positive for cannabinoids after consuming hemp-based foods. So this is how the experiment was designed. So there were two parts of this experiment, and they were both assays performed for the detection of 16 cannabinoids. And assays are just like lab procedures that analyze a substance and try to see how it behaves or what it's made of. And these are the 16 cannabinoids. Their names are really long, so these are just the abbreviations. And so the first assay was a hemp food assay on 23 hemp products. So they consisted of like sweets, beer, teas, oils, spreads, and seeds. And the purpose of this assay was to determine the concentrations of whatever cannabinoids were present in each hemp product. And then the second assay was a urine assay, and there were 22 participants. So each hemp product was assigned two participants, and then urine samples were collected 4 times. So once before each participant ate the hemp product, and then 8 hours after, 16 hours after, and then finally 32 hours after hemp product consumption. And the purpose of this assay was to see how long the cannabinoids stayed in the body and if the participant, after eating the hemp product, would fail the anti-doping test. And then the journal article went through a long process to prepare each assay, like each sample for the assays. But essentially, this was their process. So from either the hemp food sample or the urine sample, they extracted the cannabinoids and then they performed trimethylsilation. And this process is basically just to make sure that the samples don't burn when they are heated and turned into gas in the next step, which is analysis by gas chromatography and spectrometry. And I'm going to go over this process in the next slide. So this is GCMS, and so this was how the authors tried to find like what types of cannabinoids were in the hemp food or the urine test, and in what concentrations they were in. So first the sample was injected and immediately heated into a gas. So that's why this part is called the GC oven. And then a carrier gas, for example, in this experiment, helium gas was used. Um, the carrier gas will push the gas sample through this long tube, which is called a column, and the tube is lined with a chemical layer, so different components in the sample will be chemically attracted to the layer at different extents. So the molecules will make it to the end of the tube at different times, called the retention time, and this allows components of the sample to be separated, and that's what the authors wanted. They wanted the samples to be separated into the different types of cannabinoids. And then it enters the MS, where each molecule is hit by an electron beam, and the molecules will break into tiny ions. And then the lenses shoot the ions into the mass analyzer, and this will sort the ions based on their mass to charge ratio. So, in a practical sense, that just means their weight. And then once they hit the detector, the MS will create a fingerprint for each substance. And then the data system will match these fingerprints to known prints in a library to identify the substance and its concentration. So here were the results of that experiment. So 20 of the 23 hemp products contained a variety of cannabinoids, and some had very high concentration of these cannabinoids. And in four of the products, all of the 16 cannabinoids that were planned to be detected were present. So two of them were teas, and then there was an oil and a spread. And then also, prohibited cannabinoids were present in 30% of the urine samples 8 hours after consumption. So in those participants with those urine samples, if they were real athletes in competition, they all would have an unintentional violation of anti-doping regulations because the World Anti-Doping Agency has a zero tolerance policy on any cannabinoids except CBD. So as soon as the tests would detect a presence of those cannabinoids, that could result in serious consequences for the athlete. And then what was interesting in the results was that despite high cannabinoid concentration in teas, those cannabinoids were not detected in the urine test, except for CBD, which is not a problem because it's exempt from the regulations. And in fact, two of the teas were part of the four products, which included the entire spectrum of the tested cannabinoids. But in the urine test, like I already said, it only showed concentrations of CBD and no other cannabinoid. So this could be attributed to the fact that cannabinoids have low water solubility, and when you drink tea, you have to pour water on over the tea leaves. But since cannabinoids are not very water soluble, the water cannot pull the cannabinoids off the leaves very well. So it was this result was probably because cannabinoids stayed stuck to the tea bag, meaning the athlete never really swallowed them, which would be good news because they could pass the anti-doping test. And so here's my conclusion based on the analysis of the cannabinoid content and the human excretion studies. Hemp products present a real risk of triggering anti-doping rule violations, and an unintentional anti-doping rule violation is devastating for athletes because athletes can have their careers ruined by a failed anti-doping test. So first, it's devastating if they're at their peak condition for competition because they could lose time and miss big sports events like the Olympics or the World Cup. And it can also be bad for their reputation. So even if it was an unintentional failure, the media could spin the story in many ways, and athletes could lose their spots on teams. And it can have a huge financial and personal toll on the athlete as well because they could lose sponsorships or have their awards taken from them. So at the end of the journal article, the authors noted that it's important for athletes to be educated on the risk of hemp product consumption, because there's always a chance that they could fail the anti-doping test. This is the journal article, if anyone wants to read it. And that's it. Thank you for listening. And does anyone have any questions?
Thank you, Erwin, for that presentation. I really appreciate how you went into depth on all the scientific processes and explained how these doping violations can be triggered. I think it's really important that this is covered because we there have been very high profile instances of suspensions due to marijuana. Like Sha'Carri Richardson was suspended in 2021 for weed. So I think this is very important. Does anyone have any thing they'd like to share or any questions they have for Aaron?
Well, Sha'Carri Richardson, make sure we say, she came up positive, so we don't know exactly what, you know, she consumed. So we don't know what it was, whether it was she was smoking it or whether she was taking in a supplement or something of that nature. And that is always the tricky thing about these things, is that how do you come in contact with it a banned substance, or how do you come in contact with it, etc.?
Absolutely. I think um, I've heard a lot of stories about a lot about about other different banned substances where people have tried to argue that it's been an accident like consuming hemp products. Um, Aaron, do you have any did they mention any stories specifically about athletes or how this could particularly result in a violation or
The journal article didn't really talk about examples of athletes, but I will add that it's very tricky with these anti-doping tests because, for example, like hemp products, these hemp products can even be in like granola bars, so it's really tricky. Like you could eat a granola bar and then be subjected to an unintentional rule violation. But I do know another example of um, a Russian figure skater who was at the Olympics, and I think she accidentally consumed some of her grandfather's heart medication. But it was because they were sharing food and not because she actually took the medication. And that caused her to be suspended from the Olympics. So,
If there are no more comments or questions, then we will move on to our next presentation. Thank you, Aaron, for your wonderful explanation of the accidental rule violations. Um, our final intern for the presentations will be Siddhant Paruchiri. Siddhant will be presenting on the responsible use of biologics in sports medicine. Siddhant, can you please share your screen with us?
Okay, thank you for the introduction. Good evening, everyone. My name is Siddhant Paruchuri, and today I'll be presenting Moving Toward Responsible Use of Biologics in Sports Medicine by Scott A. Rodio. It was published to the American Journal of Sports Medicine in 2018. Next slide. So first, to begin, it's important to understand the therapies that are being discussed. This article focuses on biologics and regenerative medicine products used in sports medicine. Examples include stem cell products, as well as products derived from bone marrow and adipose tissue. These therapies have attracted a significant amount of attention because of their potential to augment and accelerate healing of soft tissue and their potential to regenerate tissues. This significant amount of interest allowed for the expansion of the use of biologic products in the sports medicine community. Because of this rapid growth of interest, though, questions regarding safety and effectiveness became increasingly important. And this growing interest eventually led to the greater involvement of the FDA. Next slide. Now, a major event relevant to this article happened on May 9th, 2018. The FDA filed complaints against two stem cell clinics. The clinics were marketing stem cell products without FDA approval and failed to comply with the manufacturing requirements. Not only was the FDA concerned about the unapproved treatments they were providing, they were also concerned about the reports they received where patients were undergoing severe, serious, and permanent harm due to taking these medications. Also, the increasing influence of biologics in sports medicine increased the number of regenerative medicine clinics in the US, which made it harder for regulations to be placed on each individual medication. Also, the use of these unapproved treatments was supported by very limited data, so it's largely untrustworthy. Rather than restricting uh, rather than restricting regenerative medicine entirely, the FDA developed a strategy aimed at both regulation and development. Next slide. Okay. In 2017, the FDA released a guidance document which specified its approach to regenerative medicine. The guidance document had two primary objectives. The first one was identifying and prosecuting clinics that would market and produce unapproved stem cell therapies. And second was to streamline approval of legitimate therapies that would have a sufficient scientific backing. Both of these directives or objectives protect patients and promote the development of safe, effective therapies. Next slide. Now, a big theme in that the article specifies is how the FDA classifies biologics. Products that are minimally manipulated and intended for homologous use fall under Section 361. I'll define those terms later. And when they fall under Section 361, they does not require FDA pre-market review. However, when biologic products violate these requirements or do not follow these requirements, or they are a new drug or biologic product, they fall under Section 351 and require pre-market review. Understanding this framework requires understanding of two key concepts that I mentioned before: minimal manipulation and homologous use. Next slide, please. Now, minimal manipulation refers to processing that does not alter biological characteristics of cells or tissues. For example, cell sorting and culture expansion exceed minimal manipulation, altering the biological characteristics of cells. Therefore, these methods would be under Article 351 and would require FDA pre-market review. Homologous use homologous use is when the product performs the same basic function in the recipient as it did in the donor. So, for example, the use of adipose tissues to regenerate cartilage is not homologous, and neither is the use of amniotic membrane for wound healing. These two processes also would fall under Section 351 and would require FDA pre-market review. Next slide. Now, an area of biologics that would feel the most impact from these regulations would be adipose-derived products. Products derived from adipose tissue are receiving increased regulation under FDA guidance. Generally, permitted practices when dealing with cells are rinsing, cleaning, sizing, shaping, and centrifugation solely to remove debris. However, not generally permitted practices include enzymatic digestion, cell isolation, expansion, and activation. These all modify the cell in some way. Now, because many adipose-derived products involve these types of processing methods, the ones that are not generally permitted, their regulatory status may continue to evolve as FDA guidance changes. Next slide. So the 21st Century Cures Act was signed into law in December 2016. It supported the regenerative medicine development by creating the Regenerative Medicine Advanced Therapy designation, which provided pathways designed to accelerate the approval of approved and qualified biologic products. This led to faster development of regenerative medicine therapies and demonstrated that regulatory regulations and development are not contradictory, but they are complementary to ensure the patient is safe. All right, next slide. Now, biologic therapies hold significant promise in the industry of medicine, but they also there exists a lot of heterogeneity among formulations. As a result, outcomes may be difficult to predict and regulatory challenges remain. Now, FDA guidance is expected to continue evolving and changing, and some preparations currently being used that are approved by the FDA may not be approved in the future and would require formal review. Practitioners should remain informed about ongoing regulatory developments while continuing to evaluate biologic therapies through clinical observation and reporting. Responsible use of biologics requires balancing their potential benefits with appropriate regulatory oversight. Many of these ideas extend beyond biologics and are directly relevant to the goals of this RISE internship. Next slide. Now, how is this article related to RISE or RRIPG? Now, the RRIPG mission states that it aims to reduce rugby injury incidents, improve athlete outcomes, and promote evidence-based approaches. The article, on the other hand, says biologics are being explored within orthopedic sports medicine, which could play a part into reducing rugby injury incidents, potential applications for injury treatment recovery, which would play a part in improving athlete outcomes, and the article emphasized the importance of reliable scientific evidence and regulation, which adhere to the idea of promoting evidence-based approaches. All right, next slide. And these are my references if you would like to read the article. Anyone have any questions?
Thank you for that, Siddhant. I think it's really interesting. I had a similar presentation about the possibilities for biologics in sports medicine, and I remember Dr. Lopez talking about how medical tourism has evolved because of these FDA restrictions. So it's interesting that you covered another article about that. Does anyone have any comments or questions for Siddhant? If not, thank you so much, Siddhant, for your presentation. That wraps up this week's Rugby Research and Injury Prevention Group Journal Club. Thank you all for attending.